Gametogenesis. The process of formation of gamete is called gametogenesis. Male gametes are the sperms and female gametes the ovum. Formation of sperm is called spermatogenesis and formation of ovum is called oogenesis. In this video, we are discussing the process of spermatogenesis in detail. Spermatogenesis takes place in testes. It includes two stages. They are the formation of spermatids and spermiogenesis. Formation of spermatid takes place by the following phases. Three phases are there in the formation of spermatid. They are the multiplication phase, growth phase, and maturation phase. In multiplication phase, the male germ cells or spermatogonial cells present on the inner wall of seminiferous tubules multiply by mitotic division and increase in numbers. Each spermatogonium is diploid and contains 46 chromosomes. So in the multiplication phase, the spermatogonial cells undergo repeated mitotic division to produce large number of spermatogonial cells. All these cells are diploid containing 46 chromosomes. Next is the growth phase. In growth phase, spermatogonia grow in size and form primary spermatocyte. It is diploid containing 46 chromosomes. The third phase is the maturation phase. In maturation phase, reduction division take place. That is, first and second meiotic divisions take place in maturation phase. So the primary spermatocyte undergo first meiotic division, leading to the formation of two equal haploid cells called secondary spermatocytes, having 23 chromosomes each. The secondary spermatocytes undergo second meiotic division to produce 4 equal haploid spermatids. Thus the spermatids are produced. You can see in the diagram the different phases. See, this is the phase of multiplication. Here the phase of growth, phase of maturation. In the phase of multiplication, these are diploid cells represented as 2n. These spermatogonial cells undergo repeated mitotic division and large number of spermatogonial cells are produced. Then comes the phase of growth. In the phase of growth, spermatogonial cells become large in size. It is also diploid with 46 chromosomes. Next comes the phase of maturation. In this phase of maturation, first and second meiotic divisions take place. In the first meiotic division, we can see two cells are produced here. They are having 23 chromosomes, that is, they are haploid. These cells are called secondary spermatocytes. Secondary spermatocyte undergo second meiotic division. Thus, 4 equal haploid spermatids are produced. So, the first stage is over, that is formation of spermatid. Next is spermiogenesis. The process of transformation of spermatids into functional sperms is called spermiogenesis. So, the formed spermatids are non-functional, they cannot fertilize the ovum, so they have to be transformed to functional sperm. This process is called spermiogenesis. After spermiogenesis, the sperm heads become embedded in the Sertoli cells to draw nourishment and are finally released from seminiferous tubules by the process called spermiation. So, after spermiogenesis, the sperm head become embedded in Sertoli cells to absorb nutrients. We know Sertoli cells are providing nutrients to the developing sperms. So the sperm heads are embedded in Sertoli cells to obtain nourishment. And finally, they are released from the seminiferous tubule by the process called spermiation. So spermiation is the process by which sperms are released from seminiferous tubules. Here, a sectional view of seminiferous tubule is given here. You can see the spermatogonial cells in the outer layer. Then primary spermatocyte, large cell. Then comes the secondary spermatocyte. Then spermatid. You can see the head is embedded in Sertoli cell. These are Sertoli cells. And functional sperms are produced. Collection of sperm is called spermatozoa. It's marked as spermatozoa. Next comes the hormonal regulation of spermatogenesis. Spermatogenesis starts at the age of puberty due to the increase in the release of gonadotropin releasing hormone from hypothalamus. So spermatogenesis starts at the age of puberty. Puberty means attaining sexual maturity. At this time, there is an increase in gonadotropin releasing hormone in the body from hypothalamus. Gonadotropin releasing hormone is released from hypothalamus, GnRH, we call it as GnRH. It is released from hypothalamus. As a result of which spermatogenesis starts. We can see the hormonal regulation in the form of a flow chart. First, here given hypothalamus. From the hypothalamus, gonadotropin releasing hormone is released, that is GnRH. This gonadotropin releasing hormone reaches anterior pituitary lobe, and from the anterior pituitary lobe, two types of hormones are released. They are luteinizing hormone or LH and follicle stimulating hormone or FSH. Luteinizing hormone or LH acts on Leydig cells. We have already studied Leydig cells. They are the interstitial cells found in testes. The luteinizing hormone acts on Leydig cells, and as a result of which Leydig cells secrete androgen, that is male sex hormone, mainly testosterone, that stimulates spermatogenesis. Then the other hormone, follicle stimulating hormone or FSH, it acts on Sertoli cells. We know Sertoli cells are nutrients providing cells. So when the Sertoli cells get stimulated by the follicle stimulating hormone, it secretes certain factors, that is certain chemicals are released that stimulate spermiogenesis. Thus the process of spermatogenesis is regulated by hormones. The main hormones involved in this process are gonadotropin releasing hormone or GnRH, then luteinizing hormone LH, and follicle stimulating hormone FSH. Gonadotropin releasing hormone is secreted from hypothalamus, and LH and FSH are secreted from anterior pituitary lobe. Next comes the structure of sperm. Human sperm is a microscopic structure. It is very minute, hence it is microscopic. Motile. Motile means having the ability to move. And haploid cell containing 23 chromosomes. It consists of 4 parts: head, neck, middle piece, and tail. A plasma membrane envelops the whole body of sperm. We can see the parts here. Plasma membrane, outer covering as the plasma membrane. Then this portion is the head, the enlarged portion, it is the head. Then a small region here we can see as neck. The following portion is the middle piece, and finally the tail. See the head portion. It is the enlarged end of the sperm containing the haploid nucleus. So the enlarged portion of the sperm is the head. It contains nucleus, marked here, nucleus. It contains the paternal chromosomes. 23 chromosomes are there. They are contained in this nucleus. Paternal chromosomes are present in this nucleus. At the tip of the head, a cap-like structure called acrosome is present. Here we can see a cap-like structure at the tip of the head. This structure, it is called the acrosome. So acrosome is a cap-like structure found at the tip of the head of sperm. This acrosome contains very strong enzymes that help in dissolving the egg membranes at the time of fertilization. So acrosome help in fertilization. Acrosome contains hydrolytic enzymes that help in dissolving membranes of the ovum for fertilization. Next portion is the neck. This small region is the neck. In the neck we can see two centrioles, proximal centriole and distal centriole. Proximal centriole help in the first cleavage of zygote. So after fertilization, the zygote undergoes division. The first division is initiated by this proximal centriole. Distal centriole provide base for the attachment of axial filaments. You can see two filaments passing through the middle piece up to the tail. This is here marked as axial filament. The distal centriole provide base for the attachment of axial filament. So that is the function of the two centrioles. Proximal centriole initiates cell division in fertilized ovum or zygote, and distal centriole provide base for the attachment of axial filament. Next portion is the middle piece. Middle piece contains centrally the axial filament, and around the axial filament mitochondria can be seen. It is spirally arranged around the axial filament. Mitochondria is a spiral in shape. It is arranged around the axial filament. This mitochondria provide energy for the movement and all activities of the sperm. Last portion is the tail. It helps to swim the sperm in fluid medium. A human male ejaculates 200 to 300 million sperms during a coitus. Coitus means sexual intercourse. Seminal plasma along with the sperms constitute semen. You can see certain questions and their answers. Formation of gamete is called gametogenesis. Second question, formation of sperm is called spermatogenesis. GnRH is released from hypothalamus. The cap-like structure at the tip of sperm head acrosome. The process of releasing sperms from seminiferous tubules. Spermiation. Name the hormones that regulate spermatogenesis. GnRH, LH, FSH. Number of chromosomes in primary spermatocytes and secondary spermatocytes. Primary spermatocyte contains 46 chromosomes that are diploid. Secondary spermatocyte contains 23 chromosomes haploid. What is acrosome? Mention its function. Acrosome is the cap-like structure found at the tip of the head of sperm. It contains very strong enzymes that help in dissolving the egg membranes at the time of fertilization. Name the different phases in the formation of spermatid. Phase of multiplication, phase of growth, and phase of maturation. What are the events taking place in the maturation phase? First and second meiotic divisions take place in maturation phase. What is spermiogenesis? The process of transformation of spermatid into functional sperm is called spermatogenesis. How many sperms are produced from a single spermatogonium? Answer is 4. Next, draw and label the sectional view of a seminiferous tubule. We have already given here. Refer diagram number 2. Draw and describe the structure of a sperm. That also given earlier. Refer diagram number 4. Name the centrioles in sperm and mention their function. The two centrioles found in sperm are proximal centriole and distal centriole. Proximal centriole initiates cell division in zygote. Distal centriole provide base for the attachment of axial filament. Attachment of axial filament. Prepare a flowchart showing the hormonal regulation of spermatogenesis. That is also given in diagram number 3. Give the schematic representation of spermatogenesis. That is diagram number 1. Refer. Next question. Mention the functions of GnRH, LH and FSH. GnRH stimulates anterior pituitary to secrete LH and FSH. Function of LH is LH acts on Leydig cells to produce male sex hormone testosterone or androgen. FSH or follicle stimulating hormone stimulate Sertoli cells to secrete certain factors that help in spermiogenesis. Where is the position of mitochondria in a sperm? Mention its function. Mitochondria is located in the middle piece of the sperm. It provides energy for the movement of the sperm. How many chromosomes are present in the sperm nucleus? That is, how many chromosomes are present in the sperm nucleus? Sperm nucleus contains 23 chromosomes. Then identify and label A, B, C and D. A flowchart is given here. Identify the marked parts A, B, C and D. We can see from A, GnRH is released. The gonadotropin releasing hormone is secreted from A. Hence A is the hypothalamus. Then GnRH reaches anterior pituitary. So B is anterior pituitary lobe. From the anterior pituitary lobe, FSH and LH are secreted. FSH acts on Sertoli cells. So C is Sertoli cells that produce certain factors. LH acts on Leydig cells and secrete androgens or testosterone. So D is androgen or testosterone. So this is the hormonal regulation. So that much is about the spermatogenesis. Thank you all for viewing this video. Please subscribe the channel and enable the bell button to get notification for the next video. Thank you.